answer it in real time. I am going to go ahead and pass the mic at this point to our Protect trainee from the University of Michigan, Paria Ashraf, and please go ahead and introduce Dr. Lash.
Thank you, Kristen. Good afternoon, everyone. Can you hear me? Just want to.
Yeah.
Cool. It's my pleasure to introduce our guest speaker today, Dr. Lawrence Lash. Dr. Lash is currently a professor and associate chair of pharmacology at Wayne State University School of Medicine in Detroit, Michigan, where he has been on the faculty since 1988. He received his Ph.D. in biochemistry from Emory University in 1985. After a postdoctoral fellowship at the University of Rochester, he moved to Wayne State. His research has focused on studying various aspects of metabolism, transport, and disease models that influence susceptibility to chemically induced kidney injury. Dr. Lash has published more than 120 peer-reviewed manuscripts, more than 70 reviews, and book chapters. He has also served as editor-in-chief and associate editor for prominent journals in the field of pharmacology and toxicology. Dr. Lash is also extensively involved in the education of medical and graduate students. He oversees the longitudinal coverage of pharmacology in the curriculum, runs an online clinical pharmacology elective, and provides lectures in several areas. We are very happy to have Dr. Lash today to talk more about his research, career path, and advice to trainees. I'll now hand it over to Dr. Lash. Thank you.
Well, thank you for the kind introduction. I assume I can be heard. It was working earlier, so I appreciate the opportunity to speak to everyone. Welcome to my basement office, since we're all, quarantined. Anyway, so what I'm going to do is kind of give an overview of some of the things I've done over the last, I guess, 25, 30 years in terms of focusing on trichloroethylene. And in the beginning part, I won't show a lot of actually data. It'll be more summaries to put things in context. And then I wanted to focus on some of the recent things that we're have done and hoping to do. I have a pending grant. And then at the end, I'll talk a bit about career paths, and I was asked to add that. So as I said, initially, I'm going to give sort of an overview, a bit of background on trichloroethylene. I'm sure a lot of you being involved in a Superfund research center are familiar with trichloroethylene, or TCE. So I'll talk a little bit about its properties and what it's used for, a little background, a summary of the risk assessments and regulations, and then what's important to kind of put my work in context is the existence of species-dependent differences in susceptibility, target organs, and metabolism. Then talk a bit about the metabolism, and then primarily I'll focus on the more recent mechanistic work with dichlorophenylcysteine, DCVG, which is the penultimate nephrotoxic or kidney toxic metabolite. And I'll summarize those studies and then talk more. Need to go back a second. There we go. Talk more about the human proximal tubular cell primary cultures as an in vitro model and both the advantages and limitations of that. Then I want to talk a bit about, as I said, the more recent work that we're doing on biomarkers of TCE exposure with the kidney and with DCVG in particular, using proteomic, lipidomic, and metabolomic approaches, and also some exploratory work with exosomes as a source of biomarkers. Then at the end, as was mentioned, I'm going to talk a bit about career paths, and not that I have, I think, that unique or unusual a path, but I think in terms of people who are training in toxicology and pharmacology, that gives you a lot of advantages in terms of the breadth and opportunities to contribute in different aspects of science. So, so here is the structure of trichloroethylene. A fairly simple small molecule. It's a liquid at room temperature. And it's amazingly, and I've told this story a lot. I remember when I first started working on on TCE metabolism. I had started working on the nephrotoxicity of TCE as a postdoc in the mid 80s, but when I first started working, as I'll talk about later, with the EPA on metabolism in the early to mid 90s, I had a poster at a FASEB meeting, and that was before it was called experimental biology. It was called just FASEB. And someone came up to my poster and said, "I can't believe people are still working on TCE." Yeah, I worked on this almost 20 years ago, and but so it's actually kept up a lot of us very busy. It's an interesting molecule, has a myriad, as I'll summarize, of target organs and mechanisms, and is quite interesting, as I hope you'll agree. So TCE, what it's used for. It is primarily used as a metal degreasing agent. It's also a component of paint strippers, adhesive solvent in various paints and varnishes. But because of its physical chemical properties as a liquid at room temperature, and because of its vapor pressure, it readily contaminates soil, groundwater, and surface water. It also is a volatile liquid, so it contaminates air. And it's a major component of the US EPA Superfund sites. And in fact is number 16 on the list, the substance priority list for ATSCR. So there's been a lot of interest about TCE contamination and its adverse effects for a number of years. This kind of summarizes the most recent risk assessments. The EPA, I had actually started, as I'll talk about at the end, I started working with the EPA in the early 1990s. They started working on a toxicological review of TCE, and it took them almost 20 years to publish it because there's a lot of controversy, or has been. But in 2011, they finally published their toxicological review. This is the IRIS review, which is the Integrated Risk Assessment Series. The limit for exposure in or levels in drinking water is 5 parts per billion. They also, the EPA also conducted, actually 2 weeks ago, I was part of this working group, the TSCA review, which is the Toxic Substance Chemical Act review. In 2014, the International Agency for Research on Cancer conducted a review of TCE, among other chemicals, and upgraded it then to a known human carcinogen, largely based on the sufficiency of data in humans, as well as animal data. And interestingly, the strongest human data results were for kidney cancer. And there was limited human data supporting liver cancer and non-Hodgkin lymphoma. And in 2015, the National Toxicology Program, which is part of the NIEHS, upgraded TCE to a known human carcinogen as well. But prior to that, it had been listed as a probable human carcinogen. So, let me see, wrong way. Okay. Now, interestingly, and this will kind of feed into what our focus has been on, primarily using human tissue as an investigative tool, is that TCE exhibits a number of species-dependent differences, as well as gender-dependent differences in susceptibility and target organs. So, for example, in male and female mice, you observe primarily liver and lung tumors and lymphomas. In male rats only, you see kidney and testicular tumors. In both male and female rats, you see leukemias. Now in humans, as I said, from the IARC study, the strongest data were for kidney cancer. That's at fairly high doses, however. Also liver, biliary tract cancer, non-Hodgkin lymphoma. And in terms of toxicities, that's been known for a long time. Actually, TCE up through the 1940s was used as an anesthetic agent, but then was shown to exhibit neurotoxicity. It also can exhibit liver, kidney, immunotoxicity, hematopoietic toxicity, reproductive, developmental, cardiovascular, even, I don't have on the list, dermal toxicities. So there are a number of adverse effects, and they differ depending upon species, gender, and a number of factors. So just to summarize a couple of key points at this point, to keep in mind is that TCE-induced carcinogenicity or toxicity in non-human mammalian species may be misleading due to the differences in target organ specificity. And I say misleading in terms of what it means for our alternate species of interest, humans. Now studies, however, in these species, particularly rodents, mostly rats and mice have been the non-human mammalian species used. They are informative regarding potential mechanisms or modes of action. However, considerable uncertainty exists in terms of both quantitative and qualitative uncertainties. However, studies in human-derived tissues, as well as, of course, the epidemiological studies that have been done, provide data that are more directly useful for human health risk assessment. So I'm going to give a brief overview of TCE metabolism and to make some key points. There are two main metabolic pathways, either oxidation by cytochrome P450s, which tend to, strictly speaking, be a high affinity, high capacity pathway, or glutathione conjugation, which is a low affinity, medium capacity pathway. The pathway is kind of summarized here at a very brief overview. So by cytochrome P450, you see a variety of metabolites, and I'll give a little more detail in a subsequent slide, but you see things like chloral hydrate, tri- and dichloroacetate, trichloroethanol, so on. The glutathione conjugation pathway, which is the one that I am primarily interested in for the kidney as a target organ, involves glutathione conjugation to form dichlorovinyl glutathione. It's broken down to dichlorovinyl cysteine, which is then bioactivated by either a cysteine conjugate beta lyase or flavin monooxygenase to give reactive thiol species that induce the nephrotoxicity. Now, to look at a little more detail, again, this is just, it's rather complex. You have a lot of different metabolites. This is the P450 dependent pathway, which does not seem to be relevant for the kidney in terms of adverse effects, but is relevant for a lot of the other target organs. But the species that you see, for example, the shown in the box is our metabolites and the parent compound that can be recovered in urine. So, for example, you see the parent compound, you see trichloroacetate shown here, trichloroethanol or its glucuronide, and other acids such as dichloroacetate, monochloroacetate, and oxalic acid. But in terms of the kidney, the target organ that I've primarily worked on, is the glutathione conjugation pathway. That is really the pathway of interest. And again, TCE shown here is conjugated with glutathione transferase glutathione to form dichlorovinyl glutathione shown here. This is broken down in the standard pathway to release glutamate and glycine to give you DCVG or dichlorovinyl cysteine. And that's kind of the sort of the focus point because we know, for example, as I'll explain shortly, you can inhibit further metabolism of DCVG and prevent its adverse effects. So we know that it's metabolism of this compound that is important. So DCVG can undergo mercaptorate, that's N-acetyl conjugation and mercaptorate formation, and that's recovered in the urine. Although for DCVG, it can either be deacetylated or further oxidized by CYP3A to form sulfoxide. And then, of course, the two main pathways are cysteine conjugate beta lyase or SMO to give you these reactive sulfur-containing metabolites that rearrange to cause the adverse effects. And so we've done work on that over the years. I just wanted to show this overview slide to give you an idea if we compare rat and human kidney in terms of relative rates of TCE metabolism. I should say this is actually overall metabolism because SIP, and I'm not going to go over the studies, but the human kidney has very little metabolism of TCE by the SIP pathway, whereas rat kidney has significant metabolism. But in terms of overall metabolism in the liver, primarily the liver, rat is a little faster than human. Glutathione conjugation, they're fairly comparable. The big differences come downstream where you get to the bioactivation steps. So glutathione glutathione conjugate breakdown by gamma glutamyl transferase is much faster in the rat, and cysteine conjugate beta lyase is about 10-fold more active in the rat. So, when you consider susceptibility and toxicity, data from the rat may be a little misleading if you want to make predictions for humans, and that's particularly important for a lot of the pharmacokinetic modeling. So the importance of knowing these pathways is that different target organs have different key metabolites. So for example, I mentioned that mice exhibit lung cancer. Humans do not. The reason that mice exhibit lung cancer is that they have very slow metabolism of chloral hydrate. So chloral hydrate tends to accumulate and cause proliferation and fibrosis. Also, both rats and humans exhibit liver cancer that's associated with trichloroacetate and dichloroacetate primarily. And both rats and human kidney have adverse effects associated with DCVG or DCVG sulfoxide. So there are species differences that are very critical in understanding the mode of action and in making predictions ultimately for humans. So again, to come back to the pathway, DCVG, so here we have TCE, the trichloroethylene. Conjugation occurs predominantly in the liver, but can also occur in kidney. If it occurs in the liver, you have this renal hepatic and enterohepatic circulation. Eventually, the glutathione conjugate gets back to the kidney. The kidney is very active at taking it up, breaks down to DCVG. So that's, again, our central compound of interest. Animals can have been exposed to DCVG, and we see the same kidney effects as if you use the parent compound, and you can specifically inhibit either any of these steps, either FMO or cysteine conjugate beta lyase, and you prevent nephrotoxicity or carcinogenicity in long-term exposures. We chose, in terms of mechanistic studies, to use primarily primary renal cell cultures rather than cell lines, or because some of the advantages are that the cells are more closely related to the in vivo cells. It's of course important to monitor and maintain differentiated function, whereas a lot of the immortalized cell lines are sometimes of ill-defined origin or do not maintain differentiated function. You can precisely define incubation conditions. It is possible to have extended viability up to 2 weeks. You can study cell type specific processes, and you can even set up your model in a way that maintains epithelial polarity, so you can study functions similar to what you would see in an intact kidney. So there are of course limitations, and you always keep that in mind. Primary cultures are difficult to maintain as opposed to cell lines. Almost each experiment is almost a new entity. Reproducibility, you have to have some standard that always has to be checked. You can have differentiation in culture, and you have a limited lifetime relative to immortalized cell lines. But by and large, there are a lot of types of processes that you can study. This summary table just kind of lists some types of processes that we've looked at. So for example, in an acute type of exposure where you're looking at a range of either low to high doses, concentrations, typically exposure times up to 24 hours, we've measured things like effects on intermediary metabolism, on specific enzyme activities, on mitochondrial function, active transport function, looking at levels of key metabolites, or looking at release of markers of cell injury, such as lactate dehydrogenase release, or vital dye uptake like trypan blue. In chronic exposures, where you can look at low single doses or even multiple doses up to a period of up to, say, 10 days, you can look at gene expression, macromolecular synthesis rates, which tell you a lot about cell proliferation, cell function. You can look at mutagenesis, DNA damage, look at DNA repair, as well as all the processes that you look at in the acute model. So the cells are typically grown. This is something people started doing work probably about 30 years ago, developing ways of doing primary epithelial cell cultures. And the standard approach has been to use a serum-free hormonally defined medium, because the problem is that even though we can purify specific cell populations, or I should say, strictly speaking, really highly enriched, you're always going to have some small degree of contamination, particularly of fibroblasts. And if you have serum in there, the fibroblasts grow very rapidly. It would overtake the epithelial cells. So instead, we use a serum-free hormonally defined medium. Then you have supplements such as sodium bicarbonate heaps, and then various growth factors, insulin transferrin sodium selenide, hydrocortisone, epidermal growth factor. And then what we found in our studies was that specifically adding triiodothyronine promoted the differentiating function and growth of proximal tubular cells, initially from the rat, and then we showed the same thing in human proximal tubular cells. You can use a variety of growth surfaces, collagen-coated dishes, flasks, or multi-well plates. You can use matrices such as transwell filter inserts to maintain polarity and look at processes that require the intact epithelium and the polarized epithelium. This is just a picture of human proximal tubular cells. On the left is a photomicrograph showing cells, and on the right is immunofluorescent cytokeratin staining. Cytokeratins, of course, are markers of epithelial cells. We also did an interesting study where we were looking at changes in the cells upon DCVG exposure, where we saw it's not so easy to see a loss of cytokeratin staining, but what happens, and this has been documented in a number of other models, when renal epithelial cells are adversely impacted and you get sublethal injury and then repair, regeneration, the initial cell populations are dedifferentiated, so we can see vimentin staining, and then cytokeratin staining comes back. This just summarizes some of the key properties that we validated in the human proximal tubular cells over the years. Of course, morphology, that they're generally cuboidal shaped, single nucleus with numerous mitochondria. We validated, of course, some of the key functions that make the proximal tubular cell, drug transport. It's important to both look at transport activity as well as transporter expression, because oftentimes when the renal epithelial cells, proximal tubular cells, undergo dedifferentiation or lose function, they tend to internalize plasma membrane transporters, and really, so they can be shown to be there by expression assays, but not functional. So we looked at a variety, the organic anion transporters, as well as cation transporters, things like P-glycoprotein, multi-resistant drug transporters, amino acid transporters as well. We also showed activity and expression of a broad range of drug metabolism enzymes, phase 1 enzymes like CYPs and FMOs, phase 2, the conjugation enzymes like glutathione transferases, glucuronylyl transferases, sulfotransferases. In terms of cellular energetics, that's critical function, looking at mitochondrial function and glucose intermediary metabolism. Redox status, of course, is something I've been interested in, glutathione status, and some of the responses such as lipid peroxidation. And then the response to toxicants, looking at various forms of cell death, as well as alterations in gene expression and cell proliferation. One of the important things and one of the advantages of using human-derived tissue is that humans exhibit genetic polymorphisms, and you can take this into account when in this model. So you, for example, see variations in expression of plasma membrane transporters, and I'll show a little bit of data on that, as well as drug metabolism enzymes. This can lead ultimately to altered pharmacokinetics. This has consequences for therapeutics because it means that you need, for some individuals, subpopulations, you may need a higher or lower dose to achieve the desired therapeutic effect, but in terms of toxicological consequences, it may alter the susceptibility. One example, and again, this is just a limited number of samples, but it illustrates, these are Western blots in human proximal tubular cells, primary cultures, for 3 key transporters, the organic anion transporter 1, organic anion transporter 3, and then P-glycoprotein or MDR1. And what you can see just by looking at the intensity of the blots is quite a variation just among this small number of samples, about 8 samples, 9 samples, in the level of these transporters. And so you can imagine then that the access of most therapeutic drugs are organic anions or cations, and so they're going to be, their transport renal accumulation will be influenced by these levels of carriers. Similarly, this is another set of data that just looks at the 3 glutathione transferases in the human proximal tubule, GST alpha, pi, and theta. And interestingly, GST pi, one sample, actually, well, 2 really. One had a very low level and one was undetectable. And you can see quite a range of expression levels, even in this limited number of samples. GST alpha had some level. This is just cytosol. And here, GST theta was less variable, but even still about 2-fold variability in expression level. And that's obviously going to impact drug disposition and both bioactivation and detoxification processes. So some of the mechanistic studies, and we've done these in both rat and human, but more recently, primarily human, we've identified mitochondria as early and primary intracellular sites of action, and this will come into play in our more recent studies. I'll get to it toward the end. We've again looked at, showed the important role of membrane transport and intracellular metabolism. We found that apoptosis occurs, and it tends to be a low concentration early exposure time response, whereas necrotic cell injury is more high concentration later exposure response. We've also showed gene expression changes with DCVG exposures related to cell death genes that are involved in regulation of sublethal injury and repair, DNA damage and repair, and also we see responses of either growth arrest or enhanced proliferation depending on the conditions. And I'll show a summary of some of that. First, this slide just illustrates the handling of glutathione conjugate DCVG, dichlorophenol glutathione, by the proximal tubular cell. So, and we've looked into each different aspect of this. So we see that DCVG, is some of it is filtered. So glomerular filtration gets into the luminal fluid. The rest of it that would be in the nearby perfusing capillaries renal plasma, the DCVG there can be transported into the proximal tubular cell via either organic anion transporter 1 and 3, or potentially the sodium dicarboxylate carrier. Then once in the cell, the glutathione conjugate has to be pumped out of the cell by either OP1A1 or the MRP2 or 4, because that's the enzyme that breaks it down. The gamma glutamyl transferase is only active, its active site faces outside the cell in the lumen. Then it goes breaks it down to DCVC, as I showed earlier. That's taken back up into the cell where it's metabolized. And so what we've shown is, for example, you can inhibit some of these transport processes with agents like probenecid, which is a broad organic anion transport inhibitor, and you diminish toxicity. You can inhibit gamma-glutamyl transferase, and you prevent toxicity, as well as inhibiting these different steps. So to provide a sort of a roadmap of how the renal proximal tubular cell handles the compound. So basically, and you sort of layer on top of this, as I mentioned, that you can have impacts for humans in particular. Genetic polymorphisms that affect membrane transporters. You can have enzyme induction or genetic polymorphisms that impact beta lyase or FMO activity. And then, of course, once you do form these reactive sulfur metabolites, they can elicit a variety of effects that really depends on dose and time. So they can, you can have more subtle effects that alter cellular regulatory processes that can lead to tumorigenesis. You can get repair that can lead to recovery, or sometimes you get these cycles of repair and that can lead to proliferation, that you lose regulatory control of that, or you can get to acute tubular necrosis and apoptosis. So this next slide just lists, again, then some of the specific regulatory pathways that we showed, we and others actually. Ted Locke did a study with Rick Schnellman as well that showed some of these results, some of these effects. But the MAP kinase pathway is involved, can be modulated by DCVC exposure. Also DNA repair pathways such as GAD153, which it increases DNA repair. Also P53, which can be involved in eliciting, depending upon dose and time, either growth arrest, apoptosis, or growth and repair. Also protein kinase C pathway is protective, and small heat shock protein, HSP27 in humans, it's HSP25 in rodents, is involved in actin stabilization and growth and repair. And so all these processes are impacted by DCVC. So basically, a couple of other important points in terms of the mode of action and kidney cancer is that DCVC is the only major TCE metabolite that is shown to be mutagenic, although it's not a very potent mutagen. So the mechanism of TCE-induced kidney cancer is still not completely clear, but we know that it's associated with formation of DCVC. And if you block that, you block its further, either its formation from TCE, or if you block its further metabolism that prevents the toxicity and carcinogenesis. And it's thought that there's both a direct genotoxic mechanism involving DCVC causing DNA damage, or I should say it's metabolites causing DNA damage, or it's thought that these cycles of a non-genotoxic mechanism involving repeated cycles of sublethal injury and repair leading to altered gene expression, altered regulation of cell proliferation and cell death that causes tumorigenesis. And so the precise balance between these two is probably fairly complex and is dose dependent and time dependent as well, but that hasn't been worked out. So what I wanted to, in the last main portion, talk about more recent work that we're trying to do, and then I'll show you a little more actual data. So our goal, and this involves both DCVC as one model, but other chemicals, kidney toxicants as well. The immediate goal is to identify sensitive and specific biomarkers of exposure to a broad array of environmental contaminants and specific classes of therapeutic drugs. And the key here is, of course, to identify exposure before significant or irreversible renal injury occurs. Because we know, for example, you have the standard biomarkers like serum creatinine and blood urea nitrogen for kidney function. Those typically don't significantly become elevated until there's a significant loss of kidney function and damage, and typically it's irreversible or is generally irreversible. There newer biomarkers have been identified in recent years, and that when I say recent, probably the last 10 years or so, such as KIM1, called kidney injury molecule 1, NGAL, which is neutrophil. It's a lipocalin protein, as well as others such as cystatin C that are more sensitive but are still associated with some degree of injury. The difference here is that from our data, we suggested that mitochondria are very early targets, both for DCVG as well as other types of nephrotoxic environmental pollutants, as well as certain therapeutic drugs, and that we may be able to see markers derived from mitochondria that are very early indicators of injury. So that's what we were proposing. And so the first hypothesis is that using primary cultures of human proximal tubular cells as the model, that we hypothesize that that exposure to environmentally relevant concentrations of toxicants results in interactions with mitochondria, release into the extracellular medium of specific either proteins, lipids, or altered metabolite patterns of metabolite. And the two models we're using is chemicals that we've worked on over the years, dichlorovinylcysteine as well as inorganic mercury. The second hypothesis, and I think this really broadens it, is we've also done some work with therapeutic agents, and it's well known that several classes of therapeutic drugs, such as antivirals, antibiotics, certain chemotherapeutic agents, are limited in their therapeutic use, or dose limited, due to kidney toxicity. And so we again suggest that using therapeutically relevant concentrations of these drugs, that they will modify mitochondria very early in their impact on the cell, resulting in release of specific proteins, lipids, and or altered patterns of metabolites. And the drugs that we are testing are cisplatin, chemotherapeutic agent. Polymyxin B that we've worked on is an antibiotic that's one of the last lines of defense for gram-negative resistant infections, but its limitation is kidney toxicity. And tenofovir, specifically in the disoproxyl fumarate form, it's an antiviral agent. That's an important agent. This is a summary of proteomics data that just shows the distribution of proteins that were recovered in the extracellular medium, cells exposed to DCVG. And the key point here is that a large proportion, in this case 42%, were derived from mitochondria. And there are other, of course, cellular components as well, but mitochondria play a large role. So that's at least consistent with the hypothesis that it could be a sensitive marker. This table, which I don't expect anybody to read, but if you want to go back to the recording, but I just want to show you the actual kind of data that you get from this is it just shows the identified protein, its accession number, molecular weight, and then the different conditions. So we have control at different time points, 4 and 24 hours, DCVG concentrations at 10 micromolar for 24 hours, also at higher concentration at 24 hours. And you see elevated levels that respond in a time and concentration dependent manner. And some of this data, which is really the key data, are shown here on the right, is plotted in the next figure for 6 markers. One is keratin. Now, one of the early effects besides mitochondria is that DCVG binds to actin and associated proteins and causes cytoskeletal abnormalities. So keratin type 1 shows here in panel A in the upper left, shows a time and concentration dependent increase, quite a substantial one. So we looked at, in these preliminary studies, at 10, 100, and 250 micromolar. 10 micromolar is still readily achievable at environmental exposures. 100 and 250 are higher. They've been documented in poisonings and other very unusual, more unusual exposures. So our goals will ultimately, these were just sort of pilot studies, our goals will be to look at more environmentally relevant exposures that generally span from, say, 1 to 50 micromolar at most. But here we were able to see a dose and time-dependent effect. Ribosomal protein is a dose-dependent effect and time-dependent as well. Hornerin is another cytoskeletal class of proteins, HSP90. The most interesting ones, however, sulfide oxidase is a mitochondrial protein that increased substantially, about 8-fold with 100 micromolar at 24 hours, and nicotinate nucleotide pyrophosphorylase as well. And so, so these data are consistent with mitochondria as well as other proteins being early indicators. We also did a little bit of work with a colleague at Michigan State, Gavin Reed, who now, I guess, went back to Australia a couple of years ago to look at lipids as another potential marker. And these are the type of data you see. This was looking both at the cell extract as well as the supernatant. And what we show is we get the same type of response in the supernatant as we see in the cell, which kind of validates that the use of the supernatant as a marker. The data are really summarized better in the next figure, which illustrates here, these are different lipids in cell and supernatant from cells either exposed to media, which would be control, for 4 hours, or cells exposed to 100 or 250 micromolar DCVC, which is the red and green. And what we see is essentially a lipid remodeling, where, as we see, and these are the different forms of phospholipids, and actually you can see a distinct pattern where the blue is the control, and you're seeing a change in the at the 2 different concentrations of DCVC. And basically, this is summarized here, that what we saw was that the odd number acyl chains, meaning 19.1, 17.1, and 17.0 phospholipids, were significantly increased upon DCVC exposure in both cell and supernatant. And also, one does not typically see odd numbered acyl chain length containing lipids at high proportion in mammalian cells. So this indicates a lipid remodeling in response to DCVC. But what's interesting is the lower exposure, lower concentration used, 100 micromolar, 24 hours, the cells reverted back to control, closer to control, whereas the 250 micromolar treated remain enriched in the odd number lipids. So this could be another indicator of exposure. Specific signature. So we did a preliminary study with isolating exosomes, because, as you know, many of you should know, exosomes is kind of the rage now, are vesicles that are secreted from a number of different cells, can be found in a number of different in urine, for example. Urinary exosomes is thought to be an important source of markers. And here we're using extracellular exosomes, which would be analogous to urinary exosomes in the in vivo kidney. What we did here was compare control and DCVC treated. And what we found was we verified that these were exosomes that we could isolate. There were a variety of other, but a significant number of mitochondrial derived proteins as well. So this may be another potential source of markers. And so basically, the key conclusion from this preliminary study was that we confirmed the presence of exosome, and that we recovered a significant number of proteins of mitochondrial origin, which supports the mitochondria as a sensitive site. So, so I just want to summarize some of these data, that the mechanistic data over the years, that TCE, nephrotoxicity, nephrocarcinogenicity is associated with metabolism to DCVC, which is bioactivated to a reactive phyl containing compound or sulfoxide by the cysteine conjugate beta lyase or flavin monoestrinase. We conducted studies that showed that DCVC can induce a variety of responses depending upon dose and time of exposure, and these include a whole range, including necrosis, apoptosis, or enhanced cell proliferation, growth arrest, and repair as well. And that the range is that necrotic response tends to be a relatively high dose and late response, whereas apoptosis is a relatively low dose and early response. Similarly, increased cell proliferation, where we see, for example, increased S phase stimulation of DNA synthesis, is again a relatively low dose early response as well. That's concurrently or slightly later than apoptosis, and that we identified a number of proteins associated with regulation of apoptosis, cell growth, stress response, such as the MAP kinase, protein kinase C, P53, small heat shock protein pathways. So mitochondria were identified in both in studies with rat proximal tubular cells as well as human as an early target. And then we've also, this is also known for a number of other compounds. So we took this as kind of a foundation of a hypothesis that proteins of mitochondrial origin or other molecules of mitochondrial origin could be markers, early markers of exposure. And the analogy, the model using primary cultures of proximal tubular cells is a model for what would occur, be analogous in vivo to recovery in the urine, so it could be applied clinically in a non-invasive source of a biomarker of exposure. So before I get onto the last part, which is to give a little discussion, I'm just looking over at the time on my other computer, a little discussion of career goals and pathways. Just of course want to thank people. Dave Putt worked for me for about 17 years and did a lot of the work. I had students in the lab, Sarah Heaney, Beth Horwitz, Brian Cummings, who's at Georgia now, did a lot of the early metabolism work in probably about 20 years ago now. Feng Zhu and Jing Wang were postdocs. And then of course funding, which I'll talk about in terms of career pathways, but from NIEHS, NIDDK, EPA, NIAAI, and then the recent proteomics work was done with the help of Paul Stemmer at Wayne State and Joe Caruso, who was at Wayne State, is now at University of Michigan. Again, I was asked to talk, since this is for a training program, to give some thoughts about career insights. And mine is not that unusual in that I think I've had sort of a linear path through the ranks of academia. But what I want to focus on is kind of the advantages of having a pharmacology and toxicology focus. Obviously, human health relevance is critical for getting people interested in your work and getting funding from most organizations. Although, of course, there's of course a strong interest in other models, such as zebrafish, for example, become very popular. C. elegans for a number of years. But human health relevance is important, and pharmacology and toxicology give you that. Applying it to things like risk and hazard assessment. And of course, we know there's no shortage of environmental and occupational contaminants to study. And you have diverse applications for both for toxicology and therapeutics. So this is just sort of a kind of showing my career path. So I started out, I did, and as of course I in the introduction talked about a bit, bachelor's degree. My undergraduate major was actually biology and also French literature at Case Western. Then I did a Ph.D. in biochemistry at Emory with Dean Jones, and that's where I started working on glutathione and glutathione-related processes ever since that point. I did a postdoc with Drag Anders at University of Rochester in Pharm and Tox, and then came to Wayne State now just over 32 years ago, and went through the ranks and have been there ever since. So in terms of research, my research has, obviously things evolve, you get different opportunities, but I've kind of stayed focused primarily on the kidneys, but also glutathione, sulfur, amino acid, sulfur metabolism. My dissertation work was on functions of the basolateral membrane and thiol metabolism, which involved looking at glutathione transport and oxidation. Then my postdoctoral fellowship with Drag Anders was supported by an F32 from NIEHS on nephrotoxicity of cysteine conjugates, and that's where I really first got into trichloroethylene work. Then my first grant when I became an assistant professor was at Wayne State was involved glutathione and mitochondria and toxic renal injury, where I sought to look at the roles of differences in glutathione handling, metabolism, and mitochondrial function in different kidney segments and how it determines the pattern of renal injury. Then I also got involved with the EPA in the early to mid 90s, looking at metabolism toxicity of trichloroethylene and perchloroethylene as a whole nother, very similar to TCE, but some interesting differences. Then a KO4, a career award from NADDK, where I really started developing the cell culture models. I also work with a colleague, Rudy Zallips, from Mercer University on mercury nephrotoxicity, which of course mercury is very different than organic compounds in many ways, but targets sulfur, also targets mitochondria, and has some mechanistic similarities. Then another project with the EPA looking at specifically at P450 and glutathione-dependent metabolism of TCE and PERC. Then funding from NIEHS, where we really fully developed the primary human proximal tubular cell culture model. And then out of that, developed collaborations that were funded with drug companies such as Pharmacia, Pfizer, a couple of different projects. I also had another area of interest is how pre-existing diseases impact response to toxicants. And for the kidney, one of the most prominent areas of interest is diabetic nephropathy, because diabetes, of course, as I'm sure you know, is particularly type 2 diabetes is increasing in the population and quite prominent. And diabetics, about 40% on average, develop impairment in kidney function. So, so that's one project that we had that was actually funded by the Department of Defense a few years ago. Then, go to the next slide. Did some work with Ardea Biosciences, which is another pharmaceutical company. Then had a small project with Jim Lyondike at MSU, looking at liver fibrosis and TCE. Then a project looking at polymyxin B, which you remember I mentioned was a drug, important drug for treating resistant infections, but has dose limitation of kidney toxicity, and this was a collaboration. And then another project applying the human cells to look at cytotoxicity with another company, Archaeogen. So, and then finally, I'm currently involved in a project, very different project with Al Rosenspire here at Wayne State, looking at mercury autoimmunity intolerance, and I'm looking at the kidney response part. And then, as I said, I have a pending project looking at mitochondrial biomarkers of renal injury, and which, as the little emoji clip art shows, we keep our fingers crossed. That is what I had today, and I'm glad to take any questions. And let's see.
Dr. Lash, thank you very much. This is Kristen.
Yes, hi.
I got a question from one of our Protect trainees, Anthony Sue. I am going to read it now.
Okay.
Any insight in the effectiveness of inhibition of CCBL versus FMO versus GGT versus DP in preventing toxicity? And he goes on to say, it likely depends on tissue, sex, and other factors too. Any insight would be greatly appreciated.
Sure. Yeah, that's a good question. So you're going to observe differences in how effective different targets are, depending upon if you're looking in males or females, if you're looking at if you're doing a human model like the human proximity receptor, or if you're looking at rat or mouse models, because there are such differences in the proportion of the pathway. And even in humans, because of genetic polymorphisms in transporters. So if you're targeting the transporter, for example, that takes up DCVG or DCVG into the cell, you're going to have a different degree of efficacy depending upon in humans the individual. So it's a fairly complex, I think, subject. There's so many factors involved, and there's so many different steps. So I think a lot of different there's so many factors that can modulate the effectiveness of the response. I hope that answers it.
Yeah, that's one other question. I might get to one more. I know we're running short on time. Again, from Anthony, what are the known relationships between mitochondria and odd-numbered lipids?
That's an interesting question. I really don't know. I haven't looked into that a lot. One of the things that we're also looking at in particular, besides just looking at the distribution of plasma membrane phospholipids, are looking at cardiolipins, because as you know, cardiolipins are selectively localized in the mitochondria, and their recovery in either exosomes or in extracellular medium, which would be analogous to what we recover in urine, may be an indicator of a mitochondrial. And so that's something, but we haven't gotten to that yet, and hopefully, as I said, pending application, we'll hopefully be able to pursue that in more depth. But that's a good question. I mean, certainly that's something that we want to delve into, is relating, and we're planning on delving into, is relating the more subtle cellular responses to recovery of different markers prior to significant loss of function at both a cellular and organ level.
Thank you. And Dr. Lash, one last question. If you had any piece of advice for the trainees who are graduate students and postdocs online who might be pursuing careers in toxicology, whether academic or otherwise, or pharmacology, or any of the environmental health sciences, what would you say right now in terms of what you would suggest in the ever-changing market?
Well, I think whatever one does, excuse me, whatever one pursues, you have to be something that you're interested in and passionate about. I think that's key. And I think you can look for opportunities. I think some of the opportunities I've had, they've just kind of appeared. I was fortunate because of what I was working on was of interest to people. But really, it's important, I think, to like what you're doing, be interested in it. Sometimes you do have to adapt, maybe apply new methodology that you haven't been used before, and that kind of opens up, because a lot of times I think what we can find out is limited by the method, either the sensitivity or the specificity of a method, and by applying a new method can open up new ways. So I think a lot of good avenues.
Well, great. Thank you very much. We really appreciate you taking the time for the presentation. We enjoyed it. We will post it online so that everyone can see it, and enjoy the rest of your afternoon.
Okay, well, thank you so much for all tuning in.
Okay, take care. Goodbye, everyone.